Dear students, today we'll discuss some important differences between meristematic and permanent tissues. Generally, when we define meristematic tissues, these meristematic tissues are those tissues which are in a continuous state of division. They divide continuously, and the new cells formed from the division of the meristematic cells, they differentiate. They lose the power of division, and then they attain some function. They attain particular structure, and then they are known as permanent tissues. So permanent tissues, they do not divide, they do not have capacity to divide. They are differentiated, so they perform a particular function, while as meristematic cells, they are in a continuous state of division. So generally, when we define meristematic and permanent tissues, we define them based on whether they divide or not. But the meristematic tissues and permanent tissues, they have number of differences, and today we'll discuss those differences. So first, we'll take meristematic tissue on one side and permanent tissue on one side. So capacity to divide. If we look at the meristematic cell tissues, they have the capacity to divide. They can always divide. They are always in a state of division. While as permanent tissues, under normal conditions, they cannot divide. So this is first difference. Second, these meristematic cells, they are undifferentiated. They do not perform any function. They can as they divide, as the new meristematic cells are formed, they can attain any function in future. But when they are meristematic, they do not have any particular function. They are only dividing. They are adding cells. So they are undifferentiated. While as permanent tissues, they are differentiated. They have taken functions. They carry a particular function. For example, they may take a function of photosynthesis. For example, mesophyll cells in leaves, they may become parenchyma cells and perform some function. They may become arenchyma. They may become chlorenchyma. They may become provide mechanical support and become collenchyma cells. So permanent tissues, they are differentiated, while as meristematic tissues, they are undifferentiated. Now, meristematic, whether they are will now look at whether they are simple and complex. If we look at meristematic cells, they are always simple. They are only composed of one type of cells. All the cells, they will be similar in shape. They will have same origin, and generally they perform the function of division. They will only divide. So they are simple. The cells will not be a mixture of, the tissue will not be a mixture of many types of cells, but single types of cells. While as when we look at the permanent tissues, they can be simple. For example, parenchymates tissue, for example, sclerenchymates tissue, collenchymates tissue. All these tissues are permanent tissues, and they are simple, which we have already studied. But they can also be complex. They can be complex. For example, xylem and phloem, that we also have studied, that they are composed of different types of cells. So meristematic cells, tissues, they are always simple, while as these permanent tissues, they can be simple, such as parenchyma, or complex, such as xylem. So these are the three differences. Let's move to next. Nature of cells. The meristematic cells are always living. You will see that meristematic cells are in a continuous state of division, so they are always living. While as if we look at the permanent tissues, it's not necessary that they are living. For example, if you look at non-living sclerenchyma, it is non-living cell. Sclerenchymates tissue is non-living tissue, while as parenchymates tissue is a living tissue. So permanent tissues can be living or non-living, while as meristematic tissues are always living. This is another difference. Then cytoplasm in cells. Cytoplasm in case of meristematic cells is dense. Why it is dense? Because they are always in a continuous state of division, and as such they always require different components. They require different uh, they require proteins, they require different enzymes to carry out the cell division, because to carry out cell division, the genetic material needs to be divided, and for that you need different enzymes to carry out replication. You need different for different enzymes to be available. You need transcription and translation. So all these processes are taking place inside the meristematic cells, and as such their cytoplasm is very dense. And in case of the permanent tissues, the cytoplasm may or may not be absent. For example, in case of a parenchymatous tissue, which is a permanent tissue, the cytoplasm is present. And at the same time, when you look at the sclerenchymatous tissue, when you look at sclerenchyma, the cytoplasm is absent. So in case of permanent tissues, it's not necessary whether it's not necessary that cytoplasm is present or not. And now if you look at the prominent nucleus, you will always see that the meristematic cells, they will have a prominent large nucleus, because inside the nucleus you have the genetic material, and in the dividing cells, in the meristematic cells, the genetic material, the DNA is active. It is replicating, it is transcribing, it is translating, and then you have a prominent nucleus. While as in case of the permanent tissues, it may be prominent, for example in case of parenchymatous cells, and it can be completely absent, such as sclerenchyma. Then pace of diffusion, it is rapid. Place of diffusion is rapid. The cell diffusion is rapid in case of meristematic cells, while as permanent tissues, they are completely differentiated. As such, under normal conditions they do not divide. And when you look at the size and shape of the cells, the size is small, shape is isodiametric, and lumen is large in case of the in case of the meristematic cells. While as if you look at the parenchymatous cell, the size is while when you look at the permanent tissues, you'll see size is large, shape varies. For example, in case of parenchymatous cells, it can be oval or round. And if you look at the size of the if you look at the structure of the collenchyma and the sclerenchyma, the shape may vary. It may be polygonal, and so on and so forth, and lumen is very narrow or wide. The another important difference is that the meristematic cells, they are compactly arranged. You will see that there will be no intercellular spaces between the meristematic tissues. While as when you look at the permanent tissue, for example parenchyma, you will see there are intercellular spaces, large intercellular spaces. And when you look at collenchyma, there are intercellular spaces, but they are completely less than the parenchymatous cells. And when you move to sclerenchyma, there are no intercellular spaces. So in case of permanent tissue, the cells are compactly, in case of meristematic tissue, the cells are compactly arranged. You do not have intercellular spaces. While as in case of the permanent tissue, you may have intercellular spaces, or in some cases they may be absent. Now if you look at the vacuoles are usually absent in case of meristematic cells, because they are dividing, and the nucleus is prominent, which takes the whole this takes a majority of the space in the cytoplasm. While as in case of the permanent tissue, the vacuole is present, and sometimes it pushes the nucleus towards the periphery. So vacuoles are usually present. They may be usually absent, but they may be present in some cases, in case of meristematic cells too. But vacuoles are generally present in case of the permanent tissues. They are present in the living cells, for example living permanent cells, for example parenchyma. So meristematic cells generally lack this vacuole, but in some cases it may be present. We have to remember that. But in case of the permanent tissues, it is present in parenchymatous cells, and it pushes the nucleus towards the periphery, which we have already studied. But when in some cases, in some cases of permanent tissues where you do not have cytoplasm at all, vacuoles as such are not present. So metabolic rate, it is very high in case of the the meristematic cells, because different processes are taking place inside the cell. As I already told you that replication, transcription, translation, all these processes are taking place inside the dividing cell. So as such, the metabolic rate is very high in case of the meristematic cells, while as in case of the permanent tissues it may be completely absent when the cell is dead, or completely lower as compared to the meristematic cells in case of the living permanent tissues. So thank you. These were some of the differences between the meristematic and permanent tissues, and we have already studied meristematic tissues, permanent tissues, and their types. And soon we'll be moving to next topic. And we have almost completed 75% of our syllabus through online classes and as well as offline classes. Thank you very much.